Hello to all, and welcome back to this week's episode of Biology Crash Course. Today we will be covering the topic of cell communication. Cell communication is extremely important for the survival of organisms. If cells cannot communicate, then they cannot coordinate the activities of an organism as a whole. Processes like muscle contraction, nerve movement, and the immune system are all carried out using the communication of cells. First, let's look at the basic steps of cell communication. The first step is reception. This is where a signal molecule binds with a receptor protein. More than likely, this signal molecule will also be a protein. These receptors are located in the hydrophilic and hydrophobic phospholipid bilayer of the cell membrane. These receptors are different from the typical transport protein located in the phospholipid bilayer because they do not transfer molecules in and out of the cell. Their only function is to receive signals from signal molecules in order to carry out functions in the cell. The second step is transduction. When the signal molecule binds to the receptor protein, it changes the receptor protein. This change is the catalyst to transduction. Transduction is a process by which signals transfer from the exterior of a cell to the interior of a cell. These signals must be transmitted properly in order to produce the proper response. In most cases, ATP made by the process of cellular respiration or photosynthesis is required to activate the receptor protein, just like for the tyrosine kinase receptor protein. The last step of cell communication process is the response. This step is the purpose of this whole cycle of cell communication. The response is when the signal that was initially received triggers a specific cellular response after its reception and transduction. Now that we've addressed the foundational steps of cell communication, it's now time to apply this biological process to the real world. Animals have the ability to carry out millions of unique processes all at the same time due to cell communication. Today we will look into the specific organism of octopi in order to have a greater understanding of how cellular communication affects our animal kingdom.
An octopus is a cephalopod species that lives exclusively underwater. The octopus uses their ability to camouflage as a way to conceal themselves from prey while hunting and to hide from potential predators. And they use cell communication in order to be able to do this, so let's take a look. The signal that transmits information from the brain of the octopus to the muscle of the octopus in order to complete camouflage is L-glutamate. L-glutamate is an excitatory transmitter located in the chromatophore of cephalopods. The octopus is able to release this neurotransmitter at high levels to control the contraction of their muscles. The L-glutamate is then able to bond to AMPA-like receptors in the chromatophore that help to amplify the signal using the exchange of potassium and sodium ions during transduction. Something else that can help these signals transduce are enzyme groups like kinase. The enzymes use hydrolysis to break apart the phosphate from the ATP and dephosphorylation in order to later complete phosphorylation, which is the attachment of a phosphate group to the kinase. The enzymes make this process of transduction much more efficient because they decrease the activation energy of the process, which in turn speeds up the chemical reactions in the cell. The chromatophores on the skin of the octopus are filled with pigment granules and are surrounded by radial muscles, which are innervated by the nerves that extend directly from the brain of the octopus. When the muscles of the octopus contract due to the L-glutamate neurotransmitter, the chromatophore expands, which allows for the pigment sac to show the chromatophore's color. Then, the response of this gives the octopus the ability to accurately camouflage themselves into their environment within seconds. Although most octopi are able to complete camouflage without any issues, there are some challenges that can prevent the signal from completing its pathway. One issue could be a defective AMPA-like receptor in the chromatophore that cannot properly bind to the L-glutamate signal in order to receive the neurotransmitter. And if the receptor cannot do this, then the process of transduction and response can never happen, and the octopus couldn't camouflage properly.
Another fascination that is a result of cell communication is octopi intelligence. Octopi have been found to solve mazes, complete difficult tasks, and engage in social behaviors. The process of learning in octopi begins with the signal acetylcholine. This is a neurotransmitter of the parasympathetic nervous system, playing a role in motivation, arousal, attention, learning, and memory. Acetylcholine is transported from the cytoplasm into individual vesicle carrier proteins on the membrane. The signal is transduced when the natural ligand acetylcholine binds to the muscarinic receptor, which is a subunit of the G protein responsible for physiological functions like releasing neurotransmitters. G proteins, also known as guanine nucleotide binding proteins, are a family of proteins made of amino acid chains held together by peptide bonds. These chains are folded into their secondary structure, the alpha helix, held together with hydrogen bonds. Once the acetylcholine binds to the muscarinic receptor, a subunit of the G protein releases bound guanosine diphosphate and simultaneously binds guanosine triphosphate through phosphorylation. Phosphorylation is the adding of a phosphate to a group of a molecule in order to prepare it and for a change in order to do work. In glycolysis, the breaking of sugar and cell respiration, enzymes break down glucose, releasing energy and transfer 1-phosphate from 1-ATP to glucose. The binding of GTP activates the G protein, allowing the dissociation of the A subunit to mediate specific responses. The acetylcholine ligand-gated ion channels form pores in the plasma membrane, mediating fast transmission and synapses. Acetylcholine enhances LTP in many areas, including the optic lobe. LTP is long-term potentiation that is responsible for actions of short-term memory and learning. You can almost think about LTP as an enzyme, even though it is a signal, because they have similar characteristics. Like an enzyme, LTP functions best at a certain pH, in this case, which is about 7.3. A fluctuation of pH can denature proteins, prohibiting substrates from binding to the active sites. The area in the brain that stores memories, the synaptic connections in the nerve cells, are more active in learning when there is more activity induced by LTP. Octopi have more ability to learn and behave in this advanced manner when LTP is present. If there is acetylcholine, an octopus can effectively learn and adapt, but with the lack of acetylcholine, the octopus will use nicotinic signaling in the central nervous system rather than the sympathetic nervous system.
Well, that concludes our crash course video for this week. We hope you are all cell signaling and cell communication experts. Always remember that the smallest entities can have the biggest impact, so the next time you witness an octopus camouflaging in danger or learning a new thing, think about how those actions happen and how they all relate back to cell communication. See you next week!